ভালো রে, আজকে আমিও ইউনিভার্সিটি যেতে পারিনি রে, আজকে আমি বাড়ি থেকেই ক্লাস নিচ্ছি।
ও
মানে সকালবেলা ওয়াজ নট এট অল পসিবল। আমি যেখানটায় থাকি সেখানে মারাত্মক জল জমে, আমি পুরো সেন্ট্রাল কলকাতায় থাকি।
ও আচ্ছা।
বাকিরা কি জয়েন করবে না আজকে এরকম?
স্যার পড়বে পড়বে, ওই ক্লাসে অ্যাটেন্ডেন্সের লিঙ্কের এদিক ওদিক হয়েছে আগের ক্লাসে,
হ্যাঁ
পড়বে
স্যার,
এখন স্যার।
অনেকের স্যার ওখানে ওয়েদার খারাপ তো নেটওয়ার্ক ইস্যু হচ্ছে।
একবার জিজ্ঞাসা করেন, দেন আই ক্যান স্টার্ট।
ওকে স্যার।
না, আমাকে নিয়ে 14 মানে 13 জন জয়েন করেছে। Okay. So, children, am I audible?
ইয়েস স্যার।
Yes, sir.
Now, see, children, we were talking about, as far as I remember, we were talking about the nitrogen fixation, right? So we
Yes,
were
sir.
talking about nitrogen fixation. Now, when we are talking about the nitrogen fixation, what happens? This nitrogen fixation, as I said, that can be nitrogen cycle, abiotic nitrogen fixation, and biological nitrogen fixation. Now, what happens? The free bacteria, like there are various free bacteria, and this free bacteria, like that of the Clostridium, Clostridium, and under that there are aerobic bacteria, aerobic bacteria, which is a aerobic bacteria, and Klebsiella, which is an facultative anaerobic bacteria. So it is a facultative anaerobe. Now there is another known as enterobacteria or enterobacter. This is what facultative symbiotic nitrogen fixing bacteria. Facultative symbiotic nitrogen fixing bacteria. This can be of three types. They are rhizobium, bradyrhizobium, and azorhizobium. And these are all the group of facultative anaerobes. Please write it down. Bro.
Excuse me, sir.
Yes, Baba.
Sir, Shiny is trying to join. Could you please let her in?
Okay.
She was facing some
Okay.
network issues.
Okay.
Thank you, sir.
Shivangi is there, Baba.
Okay, so then I'm telling her to
Okay.
try again.
Okay.
স্যার রাইজোবিয়ামের নিচের নামটা কি লিখেছেন?
Huh?
রাইজোবিয়ামের নিচের যে নামটা,
Bradyrhizobium. Bradyrhizobium.
আর ওটা অ্যাজোরাইজোবিয়াম।
Hmm. Can I rub it?
Yes, sir.
Now, what are the requirements of the organism for the process of the nitrogen fixation? Now, there are some requirements. Now, what are the requirements? First is the Neve genes. Neve genes are what? They are the nitrogen fixing genes, right? Next is what? Molybdenum and iron. Molybdenum and iron. Next is absorbance of nitrite and ammonia. Next, ATP molecules. Next, anoxic condition. Next, nitrogenous enzyme. Next is what? Electron donors. Children, always remember that the NIF gene encodes the encodes the enzyme nitrogenase, and this enzyme plays a vital role in the process of nitrogen fixation. In the process of nitrogen fixation. Please write it down over.
স্যার ওটা কি জিন্স লিখেছেন সার্কেল
NIF
করে?
genes, NIF, NIF genes. Ni, nitrogen, F stands for fixing. That's why nitrogen
ও আচ্ছা
fixing
আচ্ছা আচ্ছা।
genes.
So third one is absence of NO3 and NH3.
হ্যাঁ,
Thank you, sir.
welcome bro. Done, children?
Yes, sir.
Okay. Now, what happens? Just a minute. Just a minute. Bro. Now, just write a paragraph on nitrogenase enzyme. Write down the fixation of nitrogen is catalyzed by the enzyme. The fixation of nitrogen is catalyzed by the enzyme. Is catalyzed by the enzyme nitrogenase. Full stop. Nitrogenase is a complex protein. Nitrogenase is a complex protein comprising of azoferodoxin, and molybdoferodoxin, molybdoferodoxin in the ratio 2:1. In the ratio 2:1. full stop. Both the enzymes are cluster. Both the enzymes are cluster, comma. Molybdoferodoxin consists of two molecules. Molybdoferodoxin consists of two molecules of two proteins containing containing 2 molybdenum containing 2 molybdenum and 28 and 28 iron sulfur atoms. Iron sulfur atoms, full stop. Based on based on the function of molybdoferodoxin, based on the function of molybdoferodoxin, comma, it is termed as dinitrogenase, whereas azoferodoxin is termed as whereas azoferodoxin is termed as dinitrogenase reductase, dinitrogenase reductase, full stop. Since the redox potential since the redox potential of azoferodoxin of azoferodoxin or now since the potential of azoferodoxin is low, since the redox potential of azoferodoxin is low, ferrodoxin or flavodoxin, ferrodoxin or flavodoxin is believed to be electron donor. It's believed to be electron donor for its reduction, full stop. In this process, dissociated azoferodoxin in this process, dissociated azoferodoxin from molybdoferodoxin from molybdoferodoxin is reduced accepting electrons from accepting electrons from low redox potential low redox potential of ferrodoxin of ferrodoxin or flavodoxin, ferrodoxin or flavodoxin followed by ATP binding, followed by ATP binding. Full stop. Molybdoferodoxin bind nitrogen. Molybdoferodoxin bind nitrogen before forming nitrogenous complex. Before forming nitrogenous complex with the hydrolysis of ATP. With the hydrolysis of ATP. Full stop. Since azoferodoxin is one electron carrier, the reduction of nitrogen molecule, the reduction of nitrogen molecule requires 6 oxidation reduction cycle, requires 6 oxidation reduction cycles. Full stop. Nitrogenous can reduce, nitrogenous can reduce various types of substrates, various types of substrates along with nitrogen, along with nitrogen. Full stop. Write down. Nitrogenous enzyme is sensitive to oxygen. Nitrogenous enzymes are sensitive to oxygen and is prone to oxygen inhibition. And
Excuse me,
is,
sir, I
yeah,
do mispronounce nitrogenous enzyme.
are nitrogenous enzymes are sensitive to oxygen and are prone to oxygen inhibition, and are prone to oxygen inhibition. Hence maintenance of anoxygenic condition, hence maintenance of anoxygenic condition is needed, is needed. Full stop. Symbiotic organisms help in maintaining, symbiotic organisms help in maintaining anoxygenic condition, anoxygenic condition with the help of leg hemoglobin,
सर की हीमोग्लोबिन बोल रहे हैं
leg hemoglobin, leg, leg hemoglobin, leg hemoglobin being present at the root nodules, present at the root nodules of the symbiotic plant host, of the symbiotic plant host. Full stop. Full stop. Free living nitrogen fixing organism, free living nitrogen fixing organism maintains anoxygenic condition, maintains
सर फ्री
anoxygenic
लिविंग
condition. free living nitrogen fixing organisms maintain an oxygenic condition by maintaining high level of respiration. Full stop. Next paragraph, right? The presence of reduced flavodoxin and
Sir,
ferridoxin.
sir, sir. By
Yes,
maintaining
Baba.
high level of.
Respiration.
Okay, thank you, sir.
The process, sorry, the presence of reduced ferridoxin and flavodoxin as electron acceptors are critical for are critical for the
The
reduction reaction
flavodoxin as
the
presence of
presence
reduced.
of reduced flavodoxin and ferrodoxin as electron acceptors are critical for the reduction reaction of for the reduction reaction of nitrogen to ammonia. Full stop. The process of biological nitrogen fixation. The process of biological nitrogen fixation together with subsequent assimilation together with subsequent assimilation of ammonia into amino acid is highly energy extensive process is highly energy extensive process. Full stop. Molybdenum molybdenum is present in molybdenum is present in nitrate reductase and nitrogenous enzyme whereas iron is only associated with the nitrogenous enzyme structure. Full stop. Presence of nitrate
Sir,
and
with the
কোন সেন্টেন্সটা বলো বাবা?
sir, iron is only associated with.
With the nitrogenous enzyme.
Thank you, sir.
Full stop. Presence of nitrate and ammonia presence of nitrate and ammonia in the cell's environment is detrimental is detrimental to nitrogen fixation is detrimental to nitrogen fixation. Is it clear, Ava?
Yes, sir.
Okay. So that is about the enzyme nitrogenase, and that is about the mechanism. Now, what is the biochemistry of the nitrogen fixation? Now, I think you have done in your biochemistry, right? Nitrogen fixation.
Not yet, sir.
Okay,
No,
I
sir.
think you will be doing that, ma'am. Either SP ma'am or SMU ma'am will be doing that. So just an overview, just children, there are this pathway which I am not doing again, like the glycolysis, Krebs cycle, like that of the pentose phosphate pathway and electron transport chain. This is also present in your microbiology. Please study that. Okay, I am not doing the same because as ma'am, I think this portion is being taught by HOD ma'am, right?
Yes, sir.
So I think ma'am has done that. So that's why I am not repeating the same, because it will be a boring thing and I don't have that amount of time also. So that's why please study those portions which ma'am has already taught you all. Okay. Now we will talk about biochemistry of nitrogen fixation. Okay, children, just to correlate your concept of uh, biochemistry and microbiology, how the questions might be asked, that I'm just giving a sample. That according to you, you are designing a drug, right? Which stage of glycolysis you should target, or which steps of glycolysis you should target to stop the mechanism of glycolysis, and why? Any answers from you all? Please think and answer me. Let me see how much you can correlate your biochemistry and microbiology knowledge.
স্যার রিপিট করবেন একটু কি বললেন?
We can target ATP production. Anything more?
স্যার
I
রিপিট
said
করবেন?
you are designing a drug, and being a designer of drug, you want to stop the mechanism of glycolysis. What are the steps you should target for stopping the glycolytic mechanism, and why?
স্যার ফসফোরাইলেশনের স্টেপটা যদি আটকে দেওয়া যায়।
Okay, Onkit Aratrik is saying something. Two ATPs are utilized. If we stop their production, the reaction won't continue. Okay, it's partially right, but there is something more to it. See, Baba, you all have done the concept that RDS of glycolysis, right? What is RDS?
Rate determining step.
Very good. Rate determining step. What are the RDS of glycolysis? Who can tell me? The RDS of glycolysis are the irreversible steps. Now, that's why always a very common target is the irreversible step. And if we can inhibit the enzyme of the irreversible step, then what happens? The mechanism or the pathway stops, right? So that's why what happens, hexokinase, what is the uh, amino acid which is present at the active site of hexokinase, children? Children, tell me, you have done it. What is the amino acid, or which group of amino acid is present in the active site of hexokinase?
Kinase enzyme তো স্যার
হ্যাঁ হ্যাঁ হ্যাঁ,
কাইনেজ এনজাইম
কি বুঝতে পারছি না রাজশ্রী।
স্যার আপনার কোশ্চেনটা কি ছিল
I said what type of amino acid are present at the active site of hexokinase? See, the active site of the hexokinase contains the sulfur containing amino acid. And that's why they gets very quickly gets inhibited by the iodo group. So that's why the irreversible steps are the most common type of targets that are being associated for finding out any drug. So for determining any drug to inhibit the mechanism of glycolysis, it is always being targeted towards the enzymes that are controlling the irreversible step of glycolysis. So as soon as you can inhibit the enzyme controlling the irreversible step of glycolysis, the corresponding steps stops. As a result, the pathway gets totally inhibited. Is it clear, Baba?
Yes, sir.
Okay. Now, can I wrap it?
Yes, sir.
Okay. Now, what happens, as I was talking about, that in the mechanism of nitrogen fixation, N2 plus 3H2 plus 2H plus forms 2NH4 plus with a del G value of minus 39.3 kilojoules. So there is absorption of energy, liberation of energy which takes place because it is in the product side. And that's why this reaction is said to be as the exergonic process, because there is what, liberation of energy take place, right? So energy is being liberated during the process. Now what happens, and this mechanism requires a very high activation energy. Now what is activation energy, children?
So the amount of energy which will start any chemical reaction.
Okay,
স্যার দ্য মিনিমাম এনার্জি অ্যাক্টিভেশন দ্য মিনিমাম এনার্জি তাতে
right.
রিঅ্যাকশনটা মানে ইনিশিয়েট করবে
Dagoba, that is right. The terminology minimum is very important. It's a minimum amount of energy that the reactant molecule should acquire to form the product is said to be as the activation energy. Right, Baba? Any problem?
No, sir.
Just write down another short note, symbiotic nitrogen fixation. Write down symbiotic nitrogen fixation, right? Root nodules of legumes are the common dwelling site are
স্যার রুট নোডিউলস
of the legumes are the common dwelling sites for the process of nitrogen fixation. For the process of nitrogen fixation. Full stop. The formation of root nodules themselves. The formation of root nodules themselves may be induced by symbiotic bacteria.
So will you repeat from themselves?
The formation of root nodules may be induced by symbiotic bacteria or maybe or maybe or maybe by stem glands or maybe by stem glands developed individually. Full stop. Because, sorry, besides the occurrence of root nodules, besides the occurrence of root nodules mandatory, is not mandatory for nitrogen fixation, comma. Some grasses are found to be symbiotically associated symbiotically associated with the nitrogen fixing bacteria. Full stop. The nodules appear to be pink in color. The nodules appear to be pink in color due to the presence of leg hemoglobin. Due to the presence of leg hemoglobin. Comma, a molecular analog to human hemoglobin. Full stop.
So a molecular analog to
To human hemoglobin. Human hemoglobin. Full stop. Rhizobia rhizobia are gram negative alpha and beta proteobacteria that have acquired the ability that have acquired the ability to fix that have acquired the ability to fix atmospheric nitrogen atmospheric nitrogen in symbiosis in symbiosis with legumes in symbiosis with legumes. Full stop. Rhizobia stimulates the formation of nodules upon the upon the roots of the host plant. Upon the roots of the host plant. Full stop. There are two types of genes. There are two types of genes that are associated. That are associated. Number one, small NOD, nod genes, nod genes, or or the nodule forming gene, or the nodule forming gene, that is the genes that are responsible, that is the genes that are responsible for the development of nodules, for the development of nodules. And NIF genes, NIF genes, that is responsible, that is responsible for the mechanism of nitrogen fixation, that is responsible for the mechanism of nitrogen fixation. Children, answer your roll call. Don't leave because MN ma'am will be joining. She will be taking the next class. Aditi, Aditi, Ananya, Ananya, Aditi, Ananya, Ognivesh, Ognivesh, Ananya Guin,
Sir, sorry to interrupt, sir. It is MIF gene.
NIF genes, yeah. So N for national.
Okay.
Anirban,
Present, sir.
K present, Baba. Ananya Guin present.
Hi, sir.
Okay. Onubroto, Onubroto, Anushka Datta, roll number 7.
Present, sir.
Roll number 8. Roll number 8. Anushka Datta, roll number 8. Roll number 9. Aratrik present.
Yes, sir.
Roll number 10. Orina.
Present, sir.
Roll number 11. Orundhuti.
Present, sir.
Roll number 12. Oyon.
Present, sir.
Roll
It's
number
present,
30.
sir. Roll number 3 present.
Roll number 3 present. হ্যাঁ দিয়েছি তো। Roll number
Sir, roll
13.
number 1 or present আছে or mic কাজ করছে না আদিতি বিশ্বাস she's present.
Okay. Roll number 13.
Present,
Boishakhi.
sir.
Roll
Present,
number
sir.
14. Camellia.
Present, sir.
Roll number 15. Debanjana.
Present, sir.
Roll number 16. Debarati.
Present, sir.
Roll number 17. Ekta. Roll number 17. Ekta. Roll number 18. Esha. Kirti present. Moina.
Present,
Moina.
sir. Present, sir.
Okay. Manuswini. Roll number 22. Meghna. 23. Preeti.
22 মেঘনার একটু বাড়িতে কারেন্টের প্রবলেম হচ্ছে তাই জন্য জয়েন
Preeti.
করতে পারে
Preeti.
Sir, 23 Pallavi and
Oh,
24.
Pallavi. হ্যাঁ Pallavi.
Present, sir.
Preeti.
Present, sir.
Rajarshi present.
Present,
Rohan. Rohan.
Present, sir.
Sampriti. Sampriti. Sanjukta.
Present, sir.
Shanmitro.
Present, sir.
Shayoni,
Present, sir.
Shivangi,
Present, sir.
Trishti, Sumona, Surabhi,
স্যার এবার
বাবা
যারা স্যার চ্যাট বক্সে দিয়েছে তাদেরটা একটু
একটু হ্যাঁ বলে
কনফার্ম
দে তো
করে নেবো।
আমার। হ্যাঁ
রোল
Yes, sir.
নাম্বার 1 প্রেজেন্ট,
এক মিনিট দাঁড়া, জাস্ট মিনিট। রোল নাম্বার 1 প্রেজেন্ট, হ্যাঁ
তারপর 3 প্রেজেন্ট,
হ্যাঁ
তারপর 5 প্রেজেন্ট, 4 ও প্রেজেন্ট, 4 আর 5 প্রেজেন্ট, 1 3 4 5 প্রেজেন্ট।
ওকে, অনুব্রত অ্যাবসেন্ট তো?
হুম। রোল নাম্বার 8 ও প্রেজেন্ট, আনুষ্কা দত্ত।
ওকে,
তারপর রোল নাম্বার 27 সম্প্রীতি প্রেজেন্ট।
27, ওকে।
রোল
Any
নাম্বার
more?
32 সৃষ্টি চক্রবর্তী প্রেজেন্ট।
Any more?
আর তো দেয়নি কেউ।
ওকে, ঠিক আছে বাবা।
স্যার
ইয়া বাবা,
স্যার একটা কোয়েশ্চেন ছিল।
হ্যাঁ 100 বার।
স্যার ওই হেক্সোকাইনেস আপনি বললেন না যে আয়োডো মানে
হেক্সোকাইনেস, হ্যাঁ
গ্রুপ ইনহিবিট
হেক্সোকাইনেস,
করে,
হ্যাঁ হ্যাঁ হ্যাঁ
ওটা অ্যাকচুয়ালি মানে বায়োকেমিস্ট্রিতে মানে গ্লাইকোলাইসিসে ওটাতে গ্লুকোজ 6 ফসফেট ইনহিবিট করে না?
বাবু, দ্যাট ইজ, সেটা হচ্ছে সাবস্ট্রেট লেভেল ইনহিবিশন, সেটা আলাদা বাবা। দ্যাট
ও
ইজ it is helping the conversion of glucose to glucose 6 when increase in its larger amount, that is separate condition. I'm talking about any drug when targeted to the enzyme, that drug like compound is inhibiting the active site of the enzyme, like that of the competitive inhibition, and resulting in degrading the activity of the enzyme. বোঝাতে পারলাম?
হ্যাঁ স্যার, থ্যাংক ইউ, আমি একটু কনফিউজ হয়ে গেছিলাম।
না না, স্বাভাবিক ব্যাপার, ঠিক আছে, ইটস ওকে, অ্যাবসলিউটলি
থ্যাংক
ওকে।
ইউ স্যার।
হ্যাঁ, এমএন ম্যাম জয়েন করেছেন রে?
না ম্যামকে তো এখন
এমএন ম্যাম তো তখন এলো দেখলাম। এই তো
আচ্ছা
ম্যাম
হ্যাঁ
আছে,
মাঝ
মিটিং হোস্টে আছে তো।
আছে,
হ্যাঁ আমি
মোপিয়াদি
আছি
আছো? আচ্ছা,
হ্যাঁ তুই কি অ্যাটেন্ডেন্স দুটো পিরিয়ডের জন্যই দিচ্ছিস নাকি আমি আবার আইএমসিআরপিতে দেবো?
আমি একটা পিরিয়ডে দিয়েছি, আমি কি ওটা এক্সটেন্ড
আচ্ছা
করে,
ঠিক
তুমি
আছে,
করে দেবে
আচ্ছা
তাহলে
ঠিক
পরের
আছে। না
পিরিয়ডটা?
ঠিক আছে, আমি তাহলে করে
ওকে
নেবো।
ওকে, আমি তাহলে বেরোলাম, হ্যাঁ।
হ্যাঁ হ্যাঁ ঠিক আছে, ওকে।
গুড আফটারনুন ম্যাম।
গুড আফটারনুন স্টুডেন্ট।
গুড আফটারনুন ম্যাম।